Welcome everyone. Thank you for being on time. So originally it was my colleague Alina who was going to give the talk, but we had to do a bit of shuffling around, so I've taken over for her. I am joined here with Ben Moore, who is the project lead for the Ensembl outreach team. So we are in charge of giving webinars, providing training, answering help desk tickets, anything related to Ensembl. And we are also joined by Nishadi, who is the lead for the Ensembl microbes team. So she's in charge with her colleague Manuel of the Ensembl fungi, bacteria, and protist sites. Okay, so all materials are available on the Ensembl training page. So I'm just going to show you where you can find this. All you need to do is switch to a browser, which I am doing now, and go to training.ensembl.org, Ensembl without the E at the end. And let me zoom in a little bit so you can see it a little better. And if you click on the link at the very top of the page that says training materials, all of our materials are available in this first link. So webinar, introduction to the Ensembl Bacteria genome browser. So on the right-hand side, we've got a link to the presentation, although I have to say that my slides, I had to update some of the slides, so I will replace this with a newer version, but it's the same content. So if you want, you can download the presentation slides now. And underneath, at the very bottom, there at the section that says demos and exercises, if you open this tab, you can find screenshots of the demonstration I'm going to go through live on my screen later on. So if my demonstration was a bit too quick, or if you'd like to review it in your own time, you can find screenshots of what I'm going to show you live on my screen on this page here. Okay, and I believe, oh, thank you, Ben. So Ben has put the link into the chat box, so you have it handy on your screen as well. All right, so back to the presentation. Sorry. So today, I hope that at the end of the workshop, at the end of the webinar, that you'll know what the Ensembl Bacteria browser is all about, what type of data you can find on there. We're mainly going to show you the how to search and visualize genomic regions in the Ensembl Bacteria browser, and how you can add tracks to the region in detail view, and also looking at gene data. And most importantly, I'll also point out where you can find help and support if you need some later on when you're browsing Ensembl Bacteria in your own time. So just a few introductory slides of how the Ensembl project began. So in 1977, Fred Sanger and his team sequenced the very first genome, which was a bacteriophage. And this really kickstarted the era of sequencing and sequencing much more complex organisms. The very first draft of the human genome was published back in 2004. And this was a project that was started in the 1990s. And since then, we know that many, many more organisms have been sequenced. And today we can actually sequence a complete human genome in a matter of days. So we've come a very long way. And with the publication of this first draft of the human genome, Ensembl was launched in 1999 with the goal to automatically annotate the human genome and integrate it with other data and make it publicly available. But why do we need genome browsers like Ensembl? So sequencing has become much cheaper and faster, and because of that, genomic data is quickly piling up. But when sequencing data is produced, you get something like this on my slide. So just a bunch of A's, T's, C's, and G's. So if you try to read a genome without any annotation data, it would be like trying to read a book in a new language without a dictionary. So it wouldn't really mean anything to you. And this is what genome browsers are exactly there for. So genome browsers map annotations and different features such as genes or variants to genomic regions, giving the genomic region value. And what Ensembl does is it brings together information from a wide range of other databases into a single site. You may have heard of NCBI and UCSC. So these are two other genome browsers. They would have their own annotation pipelines, so some of the annotation may differ. And I also want to emphasize that Ensembl doesn't do its own sequencing. So we simply take publicly available sequencing data and we use our own pipeline to annotate the genomic data. So with this, today we have several Ensembl sites now. So a lot of genomes have been published since then. So we had to kind of extend the number of Ensembl sites. So we've got the Ensembl main site, which is dedicated to vertebrate genomes. We've also got the Ensembl genomes sister site, and this is dedicated to non-vertebrate genomes. And the Ensembl genome site is actually divided into different taxa. So we've got an Ensembl plants site, Ensembl metazoa, fungi, protists, and bacteria, which we are focusing on today. If you notice that your genome of interest is not available on either Ensembl or Ensembl genomes, it might be available in Ensembl rapid release. So I would check out that website as well. And for those who are interested, but we're not going to focus on these at all today. For those who are interested, we also have separate portals for the GRCH37 assembly, so the older human assembly, and also for the coronavirus genome. So the Ensembl Bacteria website features or includes a total of 31,332 bacterial genomes. And the type of data that we have available are gene annotation data, protein features. I'm sorry, I was supposed to delete this. So the variation data is actually not available. What I meant to say there is the variant effect predictor. So this is a tool that allows you to find out the consequences of your variants. So that is available. So just imagine this says variant effect predictor on here. We also have comparative genomics data available, so pan-taxonomic compara, including gene trees, homologs, and alignments. You can also display your own data, so you can upload your own data to visualize against a reference genome, for example. And we also provide programmatic access via our APIs. And the amazing thing about Ensembl is that it's completely open source. So you can actually find the source code of our infrastructure, of our background on GitHub. And all the data is available on our FTP site. And if you're interested in this, I will show you how you can retrieve, for example, whole genome data, like the full sequence of a bacterium via our FTP site in the demonstration. So Ensembl works in release cycles. And Ensembl is updated about every 3 to 4 months. So the current release we are in is Ensembl Genomes 58. And this was published back in January 2024. And we're expecting the next release, so Ensembl Genomes 59, to come out early May 2024. And with every update, what we do is we add any new genome assemblies or update gene sets. We also update our software. Maybe we add some new interfaces to the browser. We update the variation data, regulation data if available, so not for bacteria in this case. and also the comparative genomics data. And because Ensembl works in these release cycles, it's really important that you write down which release you're working with in Ensembl. And I will show you how you can find out whether you're in the current release or an older archive during the demonstration. So Ensembl Bacteria has several tools that you can use. So I already mentioned the variant effect predictor. This allows you to find out the consequences of your own variant, but we also have some other tools available for our bacterial genomes. So Hammer, which allows you to search your genomes for protein sequences, BLAST, sequence alignment, assembly converter, so a liftover for your data. And you can also retrieve Ensembl Bacteria data via our Perl API and REST API. And if you are interested in programmatic access to our data, please let me know, and I can put a tutorial, for example, or some help pages for that in the chat or in the Q&A. So if you work with microbiome data, so Ensembl has genome assembly data, genome annotation data. But if you work with microbiome data, you might also be interested in the Magnify resource. So this is another resource by EMBL-EBI, the European Bioinformatics Institute. And what it provides is it provides stable benchmark, version, and analysis pipelines for microbiome data. And what it does, or what you can do, is you can compare between different data sets and find out what species you have in your sample set. So Magnify provides free analyses of microbiome-derived data, which generates metagenomic protein databases and produces biospecific genome catalogs. And you can upload your own data for analysis, or you can just browse the data sets that are available on the browser. And just like Ensembl, all data are freely available to browse and to download via the website and the APIs. All right, so I'm going to move on to a demonstration now. So I'm going to show you how the Ensembl Bacteria website looks like. And we're going to explore the home page and find how you can find out more information about the available species that we have and genome assemblies. Afterwards, we're also going to look at a specific genomic region, so how you can visualize the genomic region on the browser, and look at gene and transcript information. So the example or the example species I'm going to use for visualizing a region is E. coli strain K12. Let me move to my browser, because if you want to follow along on your screen, you can go to the training page, open the region and detailed genes demo tab, and actually copy the species that we're going to look at from the website, from the training page, which I'm actually going to do. But we won't need this until later on. So that's just to keep it ready. Okay. So if you want to open, or if you want to visit the Ensembl Bacteria website, all you need to type in is bacteria.ensembl.org. So that's bacteria.ensembl.org. I'll also post it into the chat. And that's how you visit the Ensembl Bacteria website directly. Zoom in. Okay, so this is the homepage. For those who are already familiar with Ensembl, you'll notice that it's actually structured or laid out very similarly to the Ensembl main site. The only difference you'll see is the color coding up here. So bacteria has this kind of, I never know what color this is, gray, kind of looks like purple as well. Let's just say gray, this gray background here, whereas the current Ensembl main site has a blue background. But it's structured the same way. So for example, we've got our two search options here in the top left-hand corner. You can do a site-wide search. And in this gray box here, you can do a species-specific search. So I always use the species-specific search because it allows me to more efficiently search for data in Ensembl. On the right-hand side, you can see the available archive sites that are still live. So one thing I forgot to mention when I was talking about the release cycles of Ensembl is that we actually keep archives open, or we keep archived websites online for up to 5 years, and then we take them down. So what you're seeing here in this list is all the archived sites that are still available on the browser. Now, the data is not lost. So if you're, for example, if you wanted to access Ensembl Bacteria release 44, for example, you won't have an interface, you won't have a browser interface for it, but you will still have the data available to you on the FTP site. So how do I find the FTP site for Ensembl Bacteria? Very straightforward. If you go back to the browser, at the top of the page, there is a navigation bar. So these are just some quick links that to popular pages for Ensembl. You can go to downloads. I'm going to click on downloads. And once you click on downloads, you can see FTP downloads. So this is the documentation page for the FTP sites, or the FTP, the data that we have available on our FTP site. And if you scroll down, you will actually find this massive list of all the species available, including the type of data files we have available for them. So let's just open a random one. So I'm just going to open one for this very first species here. So what you can do is let's say you want to download the entire genomic sequence of Actinobacillus rosei. Just click on FASTA or FASTA format. And that leads you to the FTP site for that bacterium. For those who are unfamiliar with FTP sites, so these are structured in a very kind of very simple way. So what you're seeing here at the very top of the page is the what's the word? The path to your directory, so to your folder, sorry. And so at the moment, we can see the folder we're currently in is Bacteria 85 collection. This is the species, and the folder we're in is DNA. So we are about to download the DNA FASTA file for A. rosei. If you want, you can view all the parent directories, so all the parent folders as well. So all you need to do is just click on parent directory. And let's say you want to download the peptide sequence, you can do that here. Or if you want to explore the other species that we have available, you can go up another directory and explore them here. If you go up to the pub directory, or actually, yeah, to the pub directory. No, to the release 58 directory, you can see, you can see the different taxa we have available for Ensembl genomes. So bacteria, fungi, metazoa, plants, and protists. And if you click on bacteria, and you scroll down and go to FASTA. No, sorry, I'm in the wrong directory. What I want to show you is archive data, sorry. So what you want to do is you want to go to the very first directory, which is pub. And if you scroll down, you can see all these folders called release 1 through 58. So if you want to retrieve data from an older release and the browser, the interface isn't there anymore, you can do so by going to this pub FTP directory. And I will put this in the comment as well, in the chat, so you have a link for it if you want to have a look at the different data types we have available. You can also, so I'm back on the Ensembl Bacteria website now, still in the FTP download documentation page. You can also filter or search for your species of interest using the filter of this table. So whenever you see a table like this, you'll be able to filter down by using the text box in the top right-hand corner. Okay, let's go back to the home page. Just check the time. Right. So we've just explored how you can access the FTP page for Ensembl Bacteria. So now we're back on the home page, and we've also talked about the archive site. So if you want to find out which Ensembl release you're working with, you can see that here on the left-hand side, what's new in release 58. So currently we're in 58. And if you scroll down even more, so to the very bottom of the page, in the very left-hand corner, we can also see what release you're currently in over here. So Ensembl Bacteria release 58, published back in January 24. Okay, so let's explore the different species available in Ensembl, and different bacterial species available. So I've scrolled back up to the very top of the page, and under this gray box, you can find a link that says view full list of all Ensembl Bacteria species. So if we click that link, this is going to be the same, not the same list, but the same species that we've just looked at in the downloads page. So by default, we're only showing you the first 25 entries. If you want, you can change this to 50 or 100. And if you scroll down to the very bottom, you can see how many genome assemblies we've got available. So 31,332 bacterial genomes in Ensembl. If you want to search for a specific bacterium, you can use this filter just like in the downloads page. So let's say we want to search for that K12 for E. coli K12. So Escherichia coli, oh, I always spell it wrong, K12. And as you type it down, the list will filter. So I'm actually doing something wrong because this is actually a strain. So I have to type it in properly. 12. All right, so let's just try K12 then. Sorry, guys, I think my website is loading a bit slowly, so it took a bit of time to update that. So if you just type in the strain ID, actually, it makes it a bit easier for you. So I've just typed in K12, And you can see here in my list, we've got all of the available bacterial genomes that have K12 in their name. So most of them are E. coli here. And the one I was actually interested in was the substrain MG1655. So if you click on the species name, just waiting for Ensembl to load. So once you click on the species name, you get sent to the species information page. So that's what you're seeing here. Sorry, that's what you're seeing here. So here you can find information about what data we have available for this specific species. So we have the genome assembly, we've got gene annotation data, comparative genomics data, and for variation, you can see there is no variation database, but you can use the variant effect predictor for the species. If you want to find out more information about the genome assembly, so remember Ensembl doesn't do its own sequencing, but rather we retrieve, we import publicly available sequencing data and we apply our annotation pipelines on them. So if you want to find out more information about the assembly itself, for example, who has submitted it originally, you can go to Genome Assembly and click on More Information and Statistics. So here we can see the assembly ID. So if you click on the GCA ID, this will send you to the original submission page of the sequencing project. We've got some summary statistics such as the golden path length, the gene build provider. So gene build is a genome browser specific term, which basically means gene annotation data. So the gene annotation data or the K12 substrain MG1655 bacterial isolate was provided by the University of Wisconsin, and this was imported. We also have the number of coding genes and so on underneath here. So if you want to find out more information about the gene assembly, this is where you would go. Now going back to the gene information page, if you scroll down, for each type, for each data type, we also provide you with example regions or example pages. So you can view the karyotype, so just the entire circular genome of the species. You can view example regions, example genes, and also an example of a gene tree on here. All right, so this is how you can find information about the available species and genome assemblies. Now, let's look at more specific data. So let's look at a region still using the same substrain MG1655 of E. coli K12. And we're going to visualize the region, see how we can manipulate the view and add some tracks to it. So I'll talk more about tracks in a minute, and how you can find more information about specific genes. So let's move back to the browser. And I'm just going to switch back to the training page, because what I actually want to get are the coordinates for our example. So what we're going to do is we're going to search for these coordinates in E. coli K12, and I'm going to show you how you can visualize a genomic region using this example. So let me copy that and put this in the chat so you can follow along if you like. So since I'm on the species information page already, I can actually use this search bar here within this page. So this search bar is the same as doing a species-specific search. So all I need to type in here are my coordinates, and these are in a very particular format. So for bacteria, all you need to type in is chromosome because they only have one circular chromosome, followed by a colon, followed by the start position, followed by a hyphen, followed by an end position. Sometimes you can find commas in your coordinates or in your positions. You don't have to delete them. Ensembl will just ignore them, so you can leave them in. But in our case, these are deleted, so that's fine. So I'm just going to click on Go. Okay. And because we've searched for a specific region in, I'm just going to call it MG from now on because I can't spell out the whole number all the time. So because we've searched for a specific region in this isolate, we actually land on the location tab. So that's what you're seeing here underneath the species name. And as you look or as you search for different features, you will notice that these tabs will get populated. So currently we're just looking at a location, but once we look at a specific gene, you'll notice that there'll be a gene tab. And then if you're looking at a specific transcript, so for protein information, there'll be a transcript tab and so on. Okay, so by default, we land on the region in detail view. So that's what you're seeing here on the left-hand side in this panel. And you can switch this view to explore more information with regards to this particular region that you search for. So the region in detail page or region in detail view is made up of 3 resolutions of the genomic region that you search for. So that's what you're seeing here on the right-hand side. So first you have the chromosome view. So the region you search for is highlighted in this kind of green, in this red pie slice here. So this is just highlighting where in the genome or where in the genome your coordinates are positioned in. If you scroll down, this is the region in detail overview. So you're seeing the region you search for in this red box and some flanking regions. So you're seeing neighboring genes as well. And if you scroll down to the very bottom, this is what we call the region in detail view. So here you're only seeing the region, the coordinates that you search for, but in more detail. So what you'll notice is that these views are made up of tracks. So tracks is a genome browser specific term, and what it means is it's features mapped to a genomic coordinate, So let's focus on the genes track. So what you're seeing here is, as you can imagine, all the genes located in these coordinates. And if you click on the individual genes, so this is one gene, this block here. And if you click on a block, you can find more information about that gene. So especially when you have a lot of genes in one, a lot of genes clustered together, it's very useful to click on the box to find more information about the gene. So this particular example is a HIB-A gene, and this is a protein coding gene located on the reverse strand. And the annotation method, so how the gene was annotated, it was by ENA. And the gene GC content is 54.51. So if you wanted to see that gene in more detail, learn more about the gene itself, you could click on the gene name here. But I'm not going to do that just yet because I want to show you some other things on this view. So the nice thing about the the region in detail view is you can actually scroll across the view, and you can also highlight regions in this view. So for this, I will need to zoom out of my browser, because if you look closely in the top right-hand corner, there are actually some options that you can choose. And what this does is it changes the mouse action in the browser. So you can select a region, or you can scroll across a region. So selecting a region, all you need to do is click and drag your mouse. And once you let go of your mouse, it gives you this pop-up window, and it asks you whether you want to jump to that region or if you want to center here. So this marks the region. To scroll across, just switch your mouse action, and you can just, scroll across, and that will update the entire page, page. So this may take some time, especially if Ensembl is loading quite a lot of data at once, which is what it's doing here. So in these views, so if you scroll down to the region in detail view, in these views, you can actually configure the pages. Oh, sorry, that was a bit quick. So for example, if you wanted to add any additional tracks, or if you wanted to upload your own data, you can click on Add Remove Tracks up here for adding tracks. For custom tracks, so your own data, click on Custom Tracks. Or if you scroll up on the far left-hand side, there is another button that says Configure this page. So configure this page and Add Remove Tracks do the same thing. So you can click either. This will give you a pop-up window. And on the left-hand side, we've got some sections. So some examples of the available tracks that Ensembl has. And on the right-hand side, what you're seeing are the currently turned on, the tracks that are currently turned on. So what you could do is, for example, if you wanted to visualize the GC content in the genomic region, you can use the search option in the top left-hand corner, type in GC for GC content, and that will give you all of the tracks with GC, with the term GC in the name. If you're not sure what the track is showing you, so what information it's trying to visualize, you can just click on this eye icon on the far right-hand side, and here it just gives you a brief description. So you can turn on the track by clicking on the square, and to save your settings, just click on the check icon in the top right-hand corner. So if you're seeing something blank like this, that means that it hasn't loaded quite yet. So you just need to be patient for a couple extra seconds. So indeed, it has loaded. And if you scroll down, you'll see your new track, the GC content down here. And like spikes mean high GC content or low GC content and so on. And what you can actually do is, first of all, change my mouse action to select the region, because I actually wanted to zoom in, not scroll across. So let's just wait for Ensembl to load. So what you can do here is you can actually zoom into, for example, a high GC content region. Click and drag your mouse, jump to region. Oh, so it hasn't loaded quite yet. So this means a broken image. If you see this, just reload the page. All right. So if you zoom into, what I did was I zoomed into a high GC content region in the genome, and you can just see it in a bit more detail. I didn't quite zoom in enough. So if you zoom in a little more, you'll actually see blocks of GC content. So instead of seeing this graph, you'll be seeing boxes where there is any GC nucleotides in the genome. Okay, so I'm quite conscious about the time, so I might just skip over a few things. But one more or a couple more things that I want to show you here, especially, is how you can actually export the data in this view. So there are several ways or several formats that you can export. So first of all, you can export the image. So remember, Ensembl is completely open source, so you can use the images in Ensembl in your, for example, reports. All you need to do is cite the latest Ensembl paper, of course. So you can export the image. You can also share the page. So right now, what I did was I added a track to the view. So if I wanted to show this track to my collaborators, for example, you can generate a URL via the share button. And what you can do is you can also download the annotation of this genomic region. So if you scroll back up to the top of the page, on the left-hand side, there is a button that says Export Data. And here you can export the annotation, and you can also export the sequence of the genomic region. So in the pop-up window, under Output, you'll need to select what format you want. So do you want the sequence, or do you want to download the features? You can also highlight if you want to include any up or downstream sequences. And all you need to do is click Next. And that will, in the next page, you will be able to select whether you want to download the compressed file or the file as it is. And I always recommend to download the compressed file just because it makes it a bit faster. Okay, so how can you search for gene-specific information in Ensembl? So all you need to do, well, all that we're going to do is actually switch back to the gene information page. And to do so, I can just click on the E. coli species name up here in the top left-hand corner. And that brings me to the information page. And now using the species-specific search, I can search for a gene name. So for example, we can look for information of the LAC-Z gene. So just type in the gene name, click Go. All right, so I've searched for the LAC-Z gene in E. coli NG. I'm going to click on the gene ID. And I want you to focus on this area in the website. So you can see that we now have this new tab open called the Gene tab, and we have another tab open called the Transcript tab. So these are two very useful tabs that you're going to visit quite a lot, I would think. So in the Gene tab, what you can find is gene-specific information. So any information about the sequence of the gene, so you can visualize the sequence itself. Gene families, information about gene families, gene trees, orthologs, paralogs, and also gene functions, so gene ontology terms. Under the transcript tab, which I will switch to now. Oh, so sometimes you get this error. It actually just means that there is a kind of interruption between in talking to the server, Ensembl server. So often what helps is just refreshing the page. So as you can see, I clicked on refresh a couple times and that will load the page properly. So there was just a communication error between the servers. So when you switch to the transcript tab, This is where you can find protein-specific information. So for the gene tab is gene-specific information, and the transcript tab includes protein-specific information. So for example, if you want to highlight any, or if you want to visualize any protein domains, the protein sequence itself, we also have the AlphaFold predicted model. I'm not sure if this is available for this species, so let's just check. In this case, it's not. Yeah. So if it says error, it just means that there is no predicted model available for this. But you can view the domains and the features, for example. You can view the protein sequence. So this is just in FASTA format. Protein summary, cDNA, and so on. So I know this was quite kind of rushed the last couple of minutes. I spent a bit much time in the region and detail view. There's a lot I wanted to show you there. But one final thing I want to mention before I go through some finishing slides is where you can find help and information. So when you're in the Ensembl website, whenever you see a question mark like this, you can actually click on these, and that will give you a description of the information that you're currently seeing. So this will lead me to a documentation page regarding the gene summary. And here, in this page, you just see what information you're seeing in the gene summary view. You can also go to Help and Docs. So at the top of the page in the navigation bar under Help and Docs, if you want to find more information about specific data sets or even tutorials, you can find some helpful links under there. Okay, so just before I finish up and allow for some questions at the end of the webinar, if you did enjoy this very short, rushed webinar, and you are interested in more in-depth courses, we provide, so the Ensembl outreach team provide browser courses and REST API courses. If you are interested in these, you can go to the training page, or you can just email us directly at helpdesk@ensembl.org. If you want to reach out to us, if you have a very specific question, we are on social media. We're on Facebook, LinkedIn, X, and we also have a blog where you can find information about any updates in an upcoming release. The EBI training team have a wonderful portal for online bioinformatics training. I highly recommend visiting this very first link on my slide here. We also have Ensembl-specific tutorials, so we have them available on the Ensembl website, but also on our YouTube channel, Ensembl Help Desk, if you are interested. For any questions, any Ensembl-related questions, please feel free to email us at helpdesk@ensembl.org. So this is monitored by real people. So these aren't AI generated or anything. So if you have any questions at all, please do reach out to us. We are here to help. And if you want to sign up to any mailing list, you can do so by emailing these two emails underneath here. Now, if you are using Ensembl data, please make sure that you cite us. So this allows us to track who is using Ensembl data. If you're using Ensembl genomes data in particular, so Ensembl Bacteria, we ask you to cite the second paper here as well. So Ensembl Genomes 2022. Now, with that in mind, I just want to thank everyone who has attended here, and also all of our funders, of course, and a special shout out to Magnify. If you want to find any microbiome related data at all, Magnify is the resource to go. So we have a bit of time for some questions, any outstanding questions. So I'm going to go through them on the Q&A box.
Yeah, we still have one question. I think Nishadi is already typing in the answer. There are several number of transcripts that you find for a gene on Ensembl. So how one can select for the best transcript?
Yeah, so this is more useful for the vertebrate genomes in particular. Just in bacteria, transcription and translation happens at the same time, so there's really only one transcript there. But because of the way that Ensembl pages are laid out, we kept the kind of transcript tab there. So it's a bit misleading, but um, basically for bacteria, you'll only have one transcript. Now, if you do have multiple transcripts, for example, for Ensembl vertebrate genomes, there are some very interesting papers out there that help you decide on which transcript to select. And Ensembl does actually have a publication on that. So my colleagues have already done some work on this, and I will find the paper now and add it to the answer as well, so you have a copy of it.
Uh, there's a question about, I think, training opportunities. So there's one question where someone teaches bioinformatics tools quite regularly at workplace. So they would like to know if they can get a chance to present live or give a tutorial on some topic. So I believe Ensemble goes around the globe, I guess, providing training. So do you know if you have like collaborations with like local trainers, and are there such opportunities?
Yeah, so we actually have this workshop that we call Train the Trainer. So recently I got the opportunity to travel to Zimbabwe and train local Zimbabwe scientists how to train other people on Ensembl. So this is a very useful workshop, especially if you want to talk about Ensembl and give a tutorials yourself in there. Now, um, if it's a particular topic that you want to talk about, you can actually just email the help desk because we do provide these workshops for free. So if it's virtual, there is no cost for you at all. If it's face to face, we do ask for to pay for accommodation and transport if it's a high income country. But in general, you can just message us directly, actually, and you won't have to worry about preparing for the tutorial yourself.
Yeah. And you can also write to EMBL-EBI training team because we are always looking for trainers for many of our courses. So you can write an email with your background expertise, and then we'll get in touch with you if we are looking for a trainer. All right. So with that, this is now the full hour. So thanks very much, Luis, for the webinar today, and thanks Nishadi for joining in and helping with the questions. And thanks everyone for joining in and sending your questions. So all the best and thank you.